Welcome to the Code of Life. I'm Randine Neal. A very special next couple of episodes for you. Every year, Genome BC holds the Don Ricks Distinguished Keynote Address featuring some of BC's most spectacular and world-renowned scientists. The event is named for the late Dr. Donald B. Ricks, a BC innovator and philanthropist who was at the forefront of medicine, science, education, and the arts, and one of the co-founders of Genome BC. This year's keynote address features 3 scientists leading the fight against COVID-19 in BC. We all know Provincial Health Officer Dr. Bonnie Henry. She's joined by Dr. Mel Krajden from the BC Centre for Disease Control and Dr. Carl Hansen, CEO of AbCellera. And moderating the event, CEO of Genome BC, Dr. Pascal Spothelfer. In this episode, you'll learn about BC's biggest wins and failures in the fight against COVID-19. It's an illuminating, frank, open, and honest conversation.
This year's edition of the Don Ricks Distinguished Keynote Address is a little bit different than previous years. We may have thought that we would have been dealt lemons by forcing us to go virtual, but we have turned this into a classic case of making lemonade by having the privilege of welcoming an amazing group of panelists tonight. All 3 are doing their exceptional work right here in British Columbia, and all 3 have a profound impact on how we live through and overcome the COVID-19 pandemic. It gives me great pleasure to introduce our panelists tonight. Dr. Carl Hansen is the founding CEO of AbCellera, an industry-leading antibody discovery company poised to push therapeutic programs forward and fight disease. Until August 2019, Carl was a professor at the University of British Columbia, where he co-authored over 65 manuscripts in the fields of microfluidics, immunology, genomics, and nanotechnology. Dr. Bonnie Henry has been BC's Provincial Health Officer since 2018. She is responsible for monitoring the health of all British Columbians and undertaking measures for disease prevention and control. Dr. Henry's experience in public health, preventative medicine, and global pandemics has extended throughout her career. And Dr. Mel Krajden is the medical director of the BC Centre for Disease Control's Public Health Laboratory, medical head of hepatitis at the BC Centre for Disease Control, and a professor in the UBC Department of Pathology and Laboratory Medicine. Collectively, you are the 3 elements that are essential for our pandemic response. Mel, you and your colleagues are the early detectors and data gatherers when a public health emergency occurs. You provide the scientific evidence to inform the design of the public health response, and you support that response with ongoing research and by assuring the testing and data gathering capacity and operations. Bonnie, you then take all that data and other available information to develop the public health response and to make the critical decisions that allow us to minimize the negative population health impacts of the pandemic while balancing social and economic considerations until we have the means to deal with this virus directly. You're essentially buying the time for Carl Hansen and his team and other researchers across the world to develop the diagnostic, therapeutics, and vaccines necessary to beat the virus and allow the world to return to a more normal state of affairs, hopefully soon. Our goal today is to shed some light on each of your areas of responsibility and how you experience the unprecedented course of events this year. Following this introduction, you will provide us with remarks for 5 minutes, after which we'll engage in what I'm sure will be an animated conversation that will conclude with summarizing thoughts from each of you to close out this hour, which will be followed by an open Q&A session. So, let me please start with Dr. Mel Krajden. Mel, you and your team prepare for public health emergencies all the time. But this pandemic is obviously exceptionally impactful. Can you tell us how and when you adjusted your operations and what you did to most effectively generate the scientific evidence and supporting information to support Dr. Henry and her mandate?
Thank you, Pascal, for that introduction. So first of all, one of the roles of a public health lab, and for public health indeed, is anticipation and threat response. And in order to do that, you actually require highly qualified personnel. in the context of a laboratory, medical, scientific specialists, clinical specialists, technical and operational specialists. Those people, like a cookbook, you can't just take a cookbook and make a good meal. You have to have those people actively participating in normal operations to determine their skills, to assess the quality. And then fundamentally, if you want it to work, it has to be under what we call an academic framework, because in an academic framework, you're always thinking about improving the services. And ultimately, it's about bringing data together. It's integrating data so that you can really track the results of a test to an outcome. We knew that there were challenges in China with regard to a new agent, and we knew that in December. By January, when there was the report of this new virus, and then publicly reported the sequences of this new virus on the 10th of January, we essentially put together a in silico test that was scalable. And that test enabled us to begin testing about a week afterwards. And by the 27th of January, we were able to detect our first case. Being able to do that without depending on commercial companies gave us a lead time of about 4 to 6 weeks. And the other thing, we were nervous about this becoming a pandemic, so we actually deployed the test to a number of frontline laboratories. But we made a proviso, and that proviso is you wouldn't do the test unless you're sharing the data, because we realized that normally it would take 5 years to share the data, and you had to do it quickly. And that led to a dashboard that allowed us to track the turnaround times and the testing and who was testing. And that was a feature that we did not have in this province. Then we had the leadership of Bonnie and the government, where you actually had trust in evidence and science that helped us respond to this pandemic. And as you know, we were involved in genomics in an early phase and worked with Genome BC very quickly to try and implement genomics. So genomics allowed us to discover this agent, and then genomics allowed us to track transmissions of this agent. And with the help of Richard Harrigan and Natalie Prestigiecki, we were able to quickly show that the strains affecting British Columbians were indeed related more to the US and to Europe. And there was the first case that we recognized from Iran. That genomics is also important to understand pathogenesis. And there's been some reports about a variant, a D614G variant. And the suggestion is it is less virulent and more transmissible. But those reports are hard to integrate because you can't always show that something is less virulent unless you have good data to support it. And other people publish things that you have this deletion mutant that's less virulent. But if you look at the data carefully, you find that the people who do better are younger, and younger people do much better when they're exposed to this virus. So it really does show that to help Bonnie make her decisions, you have to integrate the data and provide it in a package. We all know we're now part of the next wave, and a challenge is test capacity. And here again, some of the efforts were to try and improve test capacity by introducing a swish and gargle technique, and this was done by Dave Goldfarb at Children's and Women's and Linda Wang at our site. And this is a way of actually making it easier to procure a sample. But again, a challenge is integrating that into the lab systems so the reports can go back to the individual as well as to the reporting structure, the medical health officers when they're positive. We also realize that there's some groups in the US that say we should test everybody all the time. And Mike Amina at Harvard has been pushing to test 4.7 million Americans every day. And that's a challenge for our systems to be able to test that at that rate. The next point is we have these newer tests that can test people early on in their infections at the point of care, and we need to figure out how to use that. You also are aware that there was serology. If we're back in April, we were thinking, oh, we could issue immunity passports. and we'll just do the serology. You've been exposed, you're immune, perfect. Well, the challenge is in fact, that immunity is not durable. There have been reinfections, and we do believe that people who've been exposed will become immune for some period of time. But what we're learning now is that every single person who seems to be responding to COVID is also responding to normal coronaviruses that are circulating and affecting humans. So why this is important is there are a bunch of vaccines that are being produced. And these vaccines, last look at the London School of Tropical Medicine, there were 248 vaccines, 49 that are in production and assessment. And the question will be, how effective are these vaccines? And are they impacted by someone's prior non-COVID corona impact? We all realize that without vaccines, we're looking at another year, maybe 2 years of ongoing infections. There's a critical value to giving the time for the life sciences in British Columbia, which is very, very strong, to be able to help us address this pandemic. And I think it's through that, the data integration and the calming voice of Bonnie Henry that will help us get there.
That's the perfect segue. So Bonnie, dealing with public health emergencies is obviously core to your job. And it's not that you haven't been dealing with very, very serious public health emergencies like the opioid crisis before the COVID-19 pandemic hit us. But the COVID-19 pandemic was exceptional in that it evolved incredibly quickly with enormous impact. So how did you experience the process of switching gears, taking whatever scientific evidence was available to you and dealing with very incomplete information about an unknown virus, and then come up with public health measures and adjust those as the scientific knowledge expanded over time?
Yeah, well, this has been, as you say, a highly unusual year, and being involved in preparedness for pandemics, infectious diseases is things that I've been involved with my entire medical career. And this year, I mean, who knew that we'd be sitting here now and that everybody out there knows what R0 is, that we know what an epidemiological curve is, that case and contact tracing are things that people understand now in ways that we hadn't even a few months ago. But we were watching very carefully early on in this. We have a group of public health experts, and Mel's part of it, people around the world who watch these things. And it's very difficult to know which of those sparks that we see out there are going to turn into something that is more concerning. We've seen it with SARS-1 in Toronto in 2003. We saw it with H1N1. And in some ways, H1N1 in 2009 gave us a sense of complacency, perhaps, because it wasn't a severe influenza pandemic in many places. And there was a sense that, oh, well, we know how to deal with this. We're not going to be hit with something that is going to cause things that are going to make us take those measures that we had been thinking about. So part of my role is to be involved in pandemic preparedness. And I took on this 2-year time-limited role of chairing the Canadian Pandemic Influenza Preparedness Task Force in 2011, and I'm still chairing it. But so I spent a lot of time looking at, what are the important things that we need to do? What do we need to learn from the past pandemics, from things that we've been through before, from the work that we've done at BC CDC and other places across the country, integrating the lab, the genomic information, the data, the scientific data, the epidemiologic data, and the communications to the public. And how do we advise government on policies around these things? And so this, one of the key things that I brought from my past experiences was the need to build in things like ethical frameworks, understanding why we would take certain measures and not others, using the principles of doing the least harm and the maximum good, and the things that are really important for us as a society when we're going through a crisis, to be upfront about, making sure that we weren't discriminating against certain groups in responding to a pandemic. So these were all the things that were going through my head. As a matter of fact, last November, I was with an international group in Rome talking about what we call non-pharmaceutical interventions to managing pandemics as part of our global pandemic preparedness. And who knew then in this wonderful fall afternoon in the beautiful city of Rome that months later it would be deserted and we'd be dealing with these issues and actually putting into force those things we were talking about. So part of my role, part of what we were doing in late December, early January, was on the phone with Mel and with others saying, what is going on? And recognizing early on that this was something that could potentially wreak havoc. We were concerned that it might be an influenza strain. There was some talk about that going around. And we were on very early on connected with the BC CDC, both for our surveillance networks that we had eyes and ears around the world talking about this, but also to get the lab ready. We knew that if we could at least have a lab test, we would be light years ahead of where we were in SARS-1 in Toronto. And I think it's a testament to that relationship we have that very early on the BC CDC was able to, within days of the genome being published, develop an in-house lab test. And that really saved us. So my role was to take all that and then to put it out to the public in ways that people understood. These were the things that we needed to do to protect each other. These were the measures that we knew would work, even though we didn't know a lot about this virus yet, and that we would learn together and to be open and upfront. And I believe, and I still believe, that if we tell people what we need them to do, and we tell them why, and we've been very upfront with presenting modeling data and all the data that we are looking at to help understand this virus and the impact of this pandemic, and we give people the means to do it, that they will do what we need. And we've shown that here in BC, that we've been able to translate that both to the politicians and the policies that we have in place from a government perspective, but also really people in BC have responded. And they've responded by doing what we need to do to bend our curve and to understand our pandemic. And we need to keep that going because we're not out of the woods yet.
Thank you, Bonnie. And Carl, I mean, talking about whirlwind and life-changing things, your team at AbCellera has gone in overdrive since you announced your focus on the novel coronavirus on January the 28th. You managed to mobilize significant financial support from the federal government. You raised venture money, and you transitioned from quote-unquote normal operations to become part of an unprecedented global effort to develop therapeutics to treat COVID-19. So can you share some of your experience over the past 10 months at the forefront of this commercial research and development efforts?
Thank you, Pascal. So it's certainly been a remarkable few months, and one that we, like everyone else, did not anticipate. I think that my comments will be directed mostly towards a lens of technology and how technology has a role to play in responding to incidents like coronavirus and things that happen in the future and in the past, as well as the diseases that we're faced with on a day-to-day basis. I lead a company, AbCellera, in Vancouver. We're a company of approximately 175 people, and we are a technology company that provides solutions to the biotech industry, working with companies large and small that are interested in searching through natural antibody responses, immune responses, to find molecules that can be repurposed as drugs. So we have spent the last 8 years building up an end-to-end platform for that problem and tackling what have been some of the hardest problems in the pharmaceutical industry. But through that work, we also engaged with a program that was funded by the Department of Defense, by the Defense Advanced Research Program Agency in the United States, that was actually focused specifically on this problem, about putting in place a technology platform that could respond quickly to a pandemic outbreak. So for a couple of years, we had been adapting our technology, which allows us to search, to analyze, to decode, and ultimately to extract antibodies from the database of immune systems to find molecules that can be repurposed as therapeutics, and making it faster and more efficient at searching through natural human antibody responses to find molecules that can be used as a therapeutic for a viral pandemic outbreak. So we had done a couple of capability demonstrations leading up to this, and this was a test of the team and an opportunity to build a network both locally and also with researchers in the United States, including some top virologists at the Vaccine Research Center at the National Institute for Health in Bethesda. As part of that work, we simulated pandemics twice. The first was on an H1N1 pandemic. The second was on a coronavirus, MERS. And we were in fact on the phone with our colleagues talking about a third capability demonstration or a test, a pressure test of this platform when the first patient was announced in the US, and that was in early January. So I was literally on the phone with our collaborators. The news came in, and within an hour we had called an all-hands meeting and decided that we would shut down all of the work in that program and in any other program that we're working on with other partners that was not urgent to focus the full weight of the company on getting ready for the coronavirus response. The first step of that process was to access a blood sample from a patient, which we were successful in obtaining from collaborators in the US just before the start of March. And on March 1st, we applied our technology to scan through that sample, and were able to identify approximately 500 antibodies from that patient. Working closely with the NIH and then with a partner, a pharmaceutical partner, Eli Lilly in the United States, we were able to take that group of 500 antibodies, winnow it down to 24 leads, and only 2 months after initiating the program, had identified the single antibody that it was decided to bring forward into clinical testing. Eli Lilly then took it from there. They're a large pharmaceutical company with the capabilities to do the manufacturing and the clinical development. And that antibody went into first-in-human testing on June 1st. So to put that in perspective, the pharmaceutical industry is many things, but it's certainly not always fast. And normally a drug development program from initiation to clinical testing would take anywhere from 3 to 5 years. This was completed in a total of 3 months, which to our knowledge is a world record in speed. And that molecule has since moved forward into 5 clinical trials, including 3 phase 2 trials and 2 phase 3 trials. And just last week, based on some interim data in the phase 3, there was a request made for emergency use authorization, which we are anxiously awaiting the result of. So this was certainly unexpected. And just to sum up the thought, I think it's a great example of why it is so important to have a vibrant sector in science that works between academia, between government and the private sector, so that we're in a position to move more quickly when situations like this emerge. And hopefully, if this has taught us anything, it's that the normal course of business, as one would put it, of taking anywhere from 3 to 5 years to get into clinical testing and 12 years to get to a product, is something that we could revise and do much better on.
Thank you, Carl. It's interesting. Let me start with a general question. Let me start our conversation with a general question. Everything mentioned, it shows that it's really a total scientific and research effort. It's not just genomics or virology. It's epidemiology, it's mathematic modeling, it's social behavioral studies, mental health research. And also the business capability to work cross-border. It's an incredible array of capabilities that come to work. And so you alluded a little bit to that, Mel. So how well equipped are we actually in BC to deal with these kind of situations? And what areas should we focus more on to actually become better? I mean, you mostly worked with US partners to advance that. So anybody wants to kind of have an opening thought on that?
I'll take a crack at that to get started. I think every jurisdiction feels like they were back on their heels on an event like this. No one was prepared for it, and in hindsight, there's always things that we could do better. The US certainly was in a better position, largely because the bulk of pharmaceutical research and biotech industry exists within their borders, and those are the tools to bring forward for the type of response that we're working on, not necessarily for the monitoring. So if anything, this has highlighted the importance of that sector, not just to the US, but internationally, that we need a strong sector. And in Canada, that we've had, not just AbCellera, but other groups in government agencies and the private sector that have contributed very meaningfully, and I would say are punching well above our weight on a per capita basis. That doesn't mean that there's not more to do, but there's there's reason to be proud, and there's also reason to double down on that and make sure that we're even better prepared for the future.
I would say I agree with you on a number of things in that, when we look at preparedness for pandemics, the US and the UK were supposed to be way up there at the top. And unfortunately, they did not do very well with their coordination around epidemiology and surveillance and monitoring, and in particular testing. And I think one of the advantages we have in BC in particular is an integration of epidemiology and the lab science, both in the BC CDC, but also in our regional health authorities, through our universities. And we're used to working with each other in a way that many other places don't have that connectivity. And we're a relatively small community, which is good in some ways, also challenging when you want to scale up in a big sense. But we have seen really the type of collaboration internationally and globally around some of these issues, particularly around the testing, around understanding what's happening in different places, supporting each other, that I've not seen ever. And we talk about it for therapeutics, but also for vaccine development. The fact that we have now at least 15 vaccines that are in phase 2 or 3 or together. And we have regulators like Health Canada speeding up their process so that it's not going to take 5 years to get something that's going to be of benefit to the population, but still maintaining those rigorous safety protocols, which I think is incredibly important.
So it's interesting. I recall meeting with Bonnie, and they said, well, there's a lot of gaps in the points that I raised for our threat anticipation and response. And, what this pandemic illustrated for me and us, I think, is that we do have many of the component pieces that enabled us to react pretty quickly. So, thanks to Bonnie and some of the approvals for data integration pieces, thanks to the Deputy Minister, thanks to the Minister of Health, we were able to actually move some of these things pretty quickly. But one of the things that you sort of see, you get on the track, and you have to learn how to adapt quickly. And to adapt quickly, we have to, I think, be even more innovative and to use better tools that are out there that we're sort of struggling with, because we have a lot of legacy systems and this has to connect to that. And we need to sometimes think out of the box. And some of that out of the box thinking comes from the kind of interactions between a vibrant life sciences sector, a fabulous public health leadership structure, and some of the investments in our educational system and our public health system.
It's interesting that you mentioned the collaboration within our ecosystem, because through the genomic enterprise, so we have parallel organizations across the country, and we see the differences between the provinces, not only from a capability point of view, because I think we're blessed having an organization like the BC CDC, which is unique in Canada, and on the other hand, the level of cooperation within the various jurisdictions is very different. And BC seems to have a higher level of collaboration. But when we then look at the Canadian response to all of it, it seems that the jurisdictional barriers between the provinces and the different approaches are really causing a lot of headaches. And you must witness that. You both must witness that on a daily basis, because it would be nice to have a bigger data set if only we could share the data with other provinces, wouldn't it?
This is probably the biggest failure coming out of SARS-1 in 2003. The Public Health Agency was created after SARS to try and support a stronger coordinated approach. And I think in one level, we certainly have that. Very early on, we created a special advisory committee that was chaired by myself and the Chief Public Health Officer of Canada. We were talking daily. We share a lot of information, but our systems are still very much fragmented. We have 13 or more health systems in this country, and it's very challenging. But I will also say with this virus, unfortunately, we are also experiencing different pandemics. They look different in different provinces, and we got hit first in long-term care, for example. But the fact that we don't have a common simple IT system that allows us to rapidly share information across the country has been one of our biggest challenges, and we should have been able to overcome that in the last number of years, but it's not got there yet. I think this is another opportunity for us to refocus on some of those important things that we do need, because we've shown that whole genome sequencing makes a difference in how we react day to day. When we had the first case in the world from Iran, we all went, well, I phoned Mel up and said, it's got to be a false positive, check everything. But we were able to find out the whole genome sequencing told us that it was indeed a different virus than the ones that we were seeing in BC. And it was a sentinel event to the world.
I would also just add the point that is integrating all of these little snippets of information into a cogent response. And I think that it's not just about genomics, it's not just about testing, it's really about thinking through the system from one end to the other and integrating the data and being able to actually have Canada function as an integrated whole could help us all, because I think it could strengthen our life sciences and our productivity in many, many areas. So
your technology to the US essentially through this was a challenge for us.
I would say we certainly haven't lost the technology. I think the reality was that we had a position within the world to respond quickly, and that borders didn't matter nearly as much as finding the right group to move that quickly to patients everywhere. So, that was the right thing to do. Of course, we are investing now on building some of those manufacturing capabilities here in Canada to have more autonomy. But coming back to Mel's point, I just wanted to, emphasize through analogy the importance of having the relationships and the communication lines in the network, whether it be between provinces or the health organizations or between the private sector and government. In our response, our technology is super fast. It's been optimized, but the big advantage was established collaborations with the big companies, with the government, with virologists, with people that could get the samples. And that, pre-existing network is something that you do not have time to make in an emergency, right? And everyone is going every different direction. If you have the relationship, you're all on the same page, you can move quickly. And so the personal factor and, getting a better line of communication, I think, is a thing that could be done in advance and that we could think about.
Yeah, I'd just add to that point, Carl, because we met with Carl talking about influenza and monoclonal antibodies. And it was through that relationship, we subsequently tried to obtain some patients who would provide their blood to enable this technology to move forward. And I think you're absolutely correct. It is those relationships that actually allow us to be more effective in all of our spheres.
And one of the things that I did early on was get a strong leadership to develop a research group network to look at what was happening with the clinical work, what was happening with around the world with therapeutics, with vaccines, with all those other pieces, and to tap into all of the different key research groups that we have here in BC. We have a number of different universities who do really great stuff. So Dr. David Patrick started that. We worked with the Michael Smith Foundation. We were able to get some early seed money to do some really basic stuff that we had prepared for, mostly thinking about influenza. And then we've built on that, and there's over $400 million worth of research being done by people in BC because of that. And we were, when CIHR and others were coming out with grants, we were well positioned here because of those relationships to actually take advantage of that.
I think that's a fantastic point. I'll just also, it's easy to lose perspective on how far we've moved since the last incident. And I'm thinking back to the SARS-1 outbreak, where it was the Michael Smith Genome Science Center that came up with the first sequence. And that was based on investments in genomics and fundamental capabilities. And it was not an easy thing to do back then. Technology has moved. Now there are many labs all over the world that can do that kind of sequencing. And here in BC, through investments from Genome BC, from CIHR, our company was formed that had a technology that could contribute to the next step of that. So, that's only, I guess that's 20 years or so. 20 years is not a long time in the arc of technology. And where will we be in another 20 years if we stay on that track, or if we even double down on those efforts?
It speaks also to the point that even if some innovation or some research outcomes are not produced here, we still need the capacity to adopt them and to work with them. And we saw that when Genome BC put in place a rapid response program to fund promising projects quickly. And that was our entire focus in that program. Within 2 weeks, we had 185 proposals. Not all of them were good, but it speaks to the fact that there is a tremendous ingenuity and research capacity that is available in BC that can jump on these things. But I think the weakness is, and you talked about the independence from commercial entities to develop a test. You go to the US to work with partners. So the industrial base we have in BC is still relatively weak to then pick up these ideas. And I guess it would be helpful to have commercial partners that could do some of the work that you need to do, Mel.
I would like to even flip that on its ear. Like you can say, you know, we have been in a position, we had leading technology in both cases. And while, you know, we don't have the big companies that can really take that next step on the global scale, we have clearly the basis from which to build those.
Yeah.
And to criticize BC for saying you don't have the pharmaceutical industry that exists south of the border is, you know, is an indictment that could be cast on every jurisdiction around the world except for the United States. So we're actually in a terrific place. And given where biotech is as a sector, the importance that's recognized in all areas of health, and now even more acutely in pandemic response, I'd say this is, you know, a great time to be thinking about how do we strengthen that position so that the next time we do have those global companies.
So I would like to just reply to one of your comments. So we had been speaking with Bonnie about, okay, how are we going to fund our genomic strategy for 5 years? And we were lucky enough to have some funded research through Genome BC. And then I remember meeting with your colleagues and talking about, well, we've got this agent that's in China and we need to think about it. And during that conversation, I had a call, and that was our first positive. And so we were having that conversation, and normally Genome BC, it's not that fast in responding to certain things. Sometimes it is, sometimes it isn't. But within a day, you said, okay, here's some seed money, begin the work. And it's that seed money that helped us get some CIHR money. So it's that relationships, it's that working together. And it's really about, I think, trying to really build on the synergies that are feasible in British Columbia. And maybe we can teach the rest of the country to do the same.
Well, and fast and slow. I mean, Carl, you mentioned that things that take 12 years are now are happening within months or under a year. So do you actually believe, and all of you, that once we have overcome COVID-19, we'll actually have learned lessons that allow us to maintain, perhaps not that speed, but certainly a different kind of pace than we had beforehand? Or will we go back to business as before, and the regulatory frameworks, etc., that we had before, and the willingness to actually cut through gone? What do you believe? Have we learned our lesson?
I hope that we've learned our lesson. I mean, there's been so much disruption over the last little while. One likes to think there's a silver lining, and the silver lining could well be that there's now, you know, rock solid evidence that the standard way of doing business in all dimensions of biomedical research can be improved. And you know, when I think about what happened with us over the last few months, the technology was a huge part of that. I do believe that we underestimate what investments in technology can do to drug development, and that it's an area that's been you know, chronically underinvested in. Ironically, because it's such an important sector, people focus on the drugs and not the process. That, of course, is what our company is setting out to do. But there are other things that are perhaps low-hanging fruit. Like we made a collaboration with our partner, Eli Lilly. That collaboration came together in 2 weeks. And that's a business transaction that normally would take a year. And it would take a year in the context of a therapeutic program trying to fight cancer. And one has to ask themselves, is cancer less urgent for those that have it than COVID? And it's not. It's just that we've become accustomed to doing business that way, and we can do much better. And certainly the other one has got to be the regulatory environment. And there's good reasons to have strict regulatory review and very high bars, but we also have to find ways to make that much faster. And going from initiation of a program to an emergency use authorization request within 7 months would have been science fiction only 12 months ago. And I don't think that will be the normal course of business in the next few years, but could we get there? Why not? That doesn't break a law of physics, and it would make a big difference to patients with all kinds of diseases.
I absolutely agree. I think we've realized that we have a lot of processes that are involved with mitigating risk, and what people think is risk could be swept away when they realize that, yes, we can actually be safe and make sure that we have the safety protocols in place without having all the bureaucratic pieces that go with it. The other part that I hope we will continue to take with us when we go forward is the importance of having Canadian basis for doing things. And we got out of the vaccine business in Canada quite a long time ago, unfortunately, and we no longer even have manufacturing capability within the country, although we're building that back up again. We didn't have the ability to manufacture PPE. We're another one of those words that everybody knows what it means now. We were dependent on China and the US, and the rise of nationalism around vaccines, around equipment that's essential for people around the world is something that I find very concerning. As much as we have spread out the knowledge, we've kept close some of the important pieces that help countries deal with this disease. So I'm hopeful that it will trigger that understanding and ongoing need to support the development that we need within the country to protect people as well.
I think that's a great point. I'll just add to that there are some of those that one needs to have some autonomy on for preparedness. And so, one could call that nationalism, but it's really readiness and independence. And there are other parts of what you just said where there's actually a strong economic incentive as well as a preparedness incentive to invest. And biotechnology in particular, you don't have to invent a case for why that would be good for the country as an economic driver, as well as a contributor to knowledge and to products that help make Canadians and others healthy. So as a sector, I think that's one of the places where the goals of being prepared and the goals of prosperity align very well. And of course, the mission of biotech is to help people. So it's one of the sectors that has all of that alignment, and one where I think BC and Canada is well positioned to grow.
So I would like to echo what you just said, Carl, because while we were able to quickly develop a test and share it in other locations, the methodology we were using was based on tried and true platforms. And then the province felt, okay, well, the solution is that we will purchase these commercial manufactured products, which actually have an end-to-end solution, put in the tube, out comes a result. And what's become clear with this global challenge is that the supply chain has been completely disrupted. And so we're back to this tried and true, less semi-automated processes. And then the need to be able to put forward those integrated technology solutions would help us right now tremendously. And yet they're absent because of the system. And so I do think that that's an important thing for us to think through, because as you point out, what's being applied today to COVID has shown us that it is destructive to the same populations who are already vulnerable and at greatest risk. And it's those populations who we need to reach out to better with technology, with support, with diagnostics, so that they can have their health inequities reduced.
So now we are in months 9 or 10 of this pandemic. And the question everybody has, okay, so what's next? How do the next 10 months look like? And there is a lot of conflicting and different views that are out there. And it's also different to sift through are statements made out of caution, out of avoiding accountability, or is it truly the best scientific understanding of where the situation is? So from a more personal point of view, How do you think the next 6 to 8 months will play out for you? Besides probably not having much vacation.
Yeah, a day off would be nice. You know what? We are in the middle of this storm. And the things that concern me right now are we are going to have to find a balance, because we know the things that we put in place where we didn't know a lot about this virus and how it was transmitting, and we knew that it can cause very exponential rapid growth and overwhelm our healthcare systems. We're not there anymore. We know a lot. We know about how to control it. We know about the things that we can do to minimize it. But it is also spreading in our community now, and we are not going to get rid of it in the next 6 months. A vaccine is going to take time. It is going to be helpful, and it's going to help us get through this. But we need to continue in that balance that we've been trying to find of controlling it in the community, protecting those people who are most at risk of severe illness and death, but also making sure for our community wellness that we have children in schools, that we have people back at work. We know the unintended consequences of what happened in March are also having effects on people's health. And as Mel said, this virus has exposed the inequities that we have in our society. And right now, we have to do everything we can to minimize those negative impacts on those people that have been impacted. And that's, women getting back into the workforce, racialized populations. We know Indigenous people have been differentially negatively affected by every past pandemic. So my focus for the next 6 months is how do we keep our community well and live with this virus, keep our schools open, keep children in classrooms, support communities to get through the next 6 to 8 months until we have a vaccine.
No.
Bonnie has highlighted the challenges that we face. I think, and I look at it from a bunch of perspectives. So one perspective is, I'm not a fierce proponent that you solve your problems by testing everybody. But I do believe having a capacity to test people, return the result very quickly so that you can modify your risks of transmitting is very important. And so we're trying to figure out the technology solutions to that, and they're not trivial because everybody else is trying to come up with the technology solutions to that. The second thing that I find very interesting is to understand the immunology of this virus. So Carl has outlined that his technology detects the antibodies to the virus. But what's very clear is that, and I mentioned this earlier, that everyone who's responding to this virus is responding to the historical viruses that they've seen in the past, the non-COVID coronaviruses. And when you think about the challenge, until you understand how those two things interact, are they interfering with the response? Are they benefiting the response? Do they lead to immunity or do they make it worse? We need to understand that because we're going to soon have an array of vaccines that we're going to need to give to our population. And we're going to need to understand whether those vaccines are safe, are they effective? And to measure that, you can think now that we need to have the tools to measure whether or not someone who got a vaccine is safe today, tomorrow, and 2 years from now, and even a lot later on. And to do that, you're going to need the data integration. So I think if we set our path now, we can solve some of these things together.
In our work, we're focused on bringing forward solutions, therapeutic antibodies, which are not a vaccine, but have a place immediately to help those that are sick or that are at high risk. And also that I believe will continue to have a place as vaccines roll out. They won't be perfect and it'll take some time. So I won't make comments that are nearly as broad as Bonnie and Mel have made, because they're actively engaged in looking at the big picture today and monitoring, what are we doing in policy and in testing to help the virus. Instead, I'll just use AbCellera as a microcosm of what everyone's doing. So we have people who are working hard on their jobs that are important, and they're trying to bring forward better therapies and technologies for coronavirus and for other diseases. And we, like everyone else, are doing that in the backdrop of constraints of distancing and dealing with constraints at home and the stresses. And one of the things that's been heartening for me is to see how resilient the team has been, and how they have made a lot of personal sacrifices to bring forward a solution that now is on the verge of actually making a difference for thousands, maybe even more people moving forward. So that's something that we're fiercely proud of. We're going to continue to do what we can, and I think that is not an exception in our community. I think there are groups like that in the private sector, in government, in academic labs that are all making do. And if we do that, that's going to be one of the more important things, as Bonnie said, to protect our communities, but also to make sure that we're not losing sight of the other factors that have unintended consequences in economics and social.
Yeah, and it's also interesting for us as an ecosystem builder and as a funder of research and innovation on how we can contribute to that ongoing effort. Because I remember we were saying, okay, so let's help research to get off the ground quickly. But then the question was, how long do we do that? At what point is kind of initiating research kind of too late to respond to that pandemic? And at the same time, looking at it now, we're going, this is not over soon. So there is a lot of work that still needs to be done. So if you look at agencies like ours, we have certain means to fund things, government and other research support organizations. What kind of things would you kind of tell me we should do?
Great question. You put me on the spot a little bit. I wasn't prepared for that one. I like to think that where the biggest value lies is normally in working on the important thing that not everyone is working on. And so I think there is a very interesting challenge here that there are so many obvious areas where we need more work in COVID. And someone needs to be doing that, but many of those are highly subscribed, both locally and around the world. And I think I would pull back a little bit and ask the more general questions, like what is the fundamental scientific question that would not just be applicable to COVID, where there is an immediate need, but that would then allow me to pivot into another area or apply that new knowledge into a new sector. And those general questions and capabilities tend to be some of the most productive things for academia to work on. And when they get to be very focused, normally private companies are better positioned to really execute on that. And when there is a problem as urgent as COVID, you can be sure that they are. So I think I would pull back a little bit and ask the bigger questions that are trying to get a step ahead of where we are today. That's not much of an answer.
No, actually, it is an answer.
On a very micro scale, there are some really important things that we continue to learn from things like using whole genome sequencing. And at first, it was very slow. It took us quite a long time. Our aim is to sequence every genome of every positive case that we have here. But it's hard to underestimate how much it changed our focus. It was knowing that we were getting flooded. We had 2 super spreader events, and it was the whole genome sequencing that supported us in understanding that. And we had one which was a dental conference, which spread to hundreds of people. The other one was getting flooded from Washington State. And it was really what enabled me to go to the Premier and the Minister and say, we need to do this now today to stop people from traveling to Washington State to make these changes, because this is what we're seeing. And so it's, we give seed support, but then we need ongoing support through this crisis right now to keep these important measures going. We're using it now to help us understand outbreaks that are happening. Are they all the same, or is it being introduced from different places? And it's a new way of thinking that helps us do epidemiology in a different way that has been extremely helpful here. And I think we're the only ones in the country that are using it that way.
I think both of those fit so well together, because the point that Carl highlights is the fact that we're all stressed out with COVID, and it's going to hang around, it's going to drive us nuts. But while COVID is going on, there's salmonella outbreaks. There's climate change, which people don't believe is occurring, but that climate change is going to affect how the environment, the food supply, and we're going to need to use genomics to better understand what are the threats to us as a society. And so when you think about the things that need to be done, it's really about, can you sequence it? When do you need to sequence it? Can you integrate the data? Can you create that package that gives Bonnie and her team the information to say, oh, we have a threat from this source, it's a food source. Can we respond to it and reduce the risks to British Columbians and other people? And then, as you pointed out at the very beginning, you need to share it, because the food supply we get is from various parts of the world. It affects us, it affects those in Ontario. You need to share the data. You need to figure out those pieces. And then you have the technology sector, and they can make it sing. And that's what we need. So you need to figure out how to do that.
Yeah, and I think we have only a few minutes left in our conversation before we move over to the Q&A. But I have to say, kind of reflecting on our conversation and going back to what this event is, it's Don Rick's Distinguished Keynote, and listening to you and seeing how BC has responded to that amazing event. I think one person would be proud about the response, be it from a technology, from an entrepreneurial, from a company point of view, be it from a public health leadership point of view, and how a depoliticized response was possible in this province. And I mean, talking labs, having that infrastructure available to provide the information that allows us, or that allows you to make these critical decisions, and to see that collaborative effort within our community, not just within silos, but across silos and between academia, industry, and the public sector, has been actually a fabulous experience. And that's kind of the silver lining that you mentioned, Carl. And I think our responsibility, I was actually afraid of your answers, Mel, because I was expecting a whole wish list of things that we could do for you. But we as a funding agency and as an ecosystem builder have really to build on that and take that strategic approach that you mentioned, Carl, and look at the gaps and look at them strategically in consultation with our stakeholders, but also respond tactically to the immediate requests and requirements that we have in the system.